The idea that yogurt could play a role in healing the brain after traumatic injury might sound like nutritional optimism gone too far, until you take a microscope to it. Specifically, a nanoparticle level microscope. Within yogurt lie extracellular vesicles, EVs, nanoscale lipid-bound packages secreted during fermentation, carrying a potent mix of proteins, lipids, RNAs, and metabolites. In preclinical models, these dairy-derived EVs are now being studied not just for gut health or immune modulation, but for something much more ambitious: repairing the brain after trauma. At first glance, it seems improbable. The blood-brain barrier, BBB, is notoriously selective, designed to shield the central nervous system from pathogens, toxins, and most therapeutics. But certain EVs, including those derived from dairy, have shown a surprising ability to cross this barrier. Their small size, typically 30 to 150 nanometers, lipid composition, and resemblance to endogenous cellular vesicles allow them to navigate the tight junctions of the BBB without triggering immune alarm bells. Once past this neurological border control, the EVs can deliver bioactive molecules to neurons, glial cells, and vascular tissue, potentially influencing brain repair from the inside. In animal models of traumatic brain injury, TBI, and ischemic stroke, early results are promising. Yogurt-derived EVs appear to reduce neuroinflammation, stabilize the blood-brain barrier itself, and promote neurogenesis and synaptic repair. Their cargo includes microRNAs like miR-148A and miR-21, known regulators of inflammation and apoptosis, as well as antioxidant enzymes and bioactive peptides that support cell survival and mitochondrial function. In rodent models, administration of dairy EVs post-injury has been associated with improved behavioral outcomes, reduced glial scarring, and enhanced cognitive performance during recovery tasks. The mechanism here is elegantly multi-targeted. These EVs don't just tamp down inflammation, though they do that well, by reducing microglial activation and dampening NF-B signaling. They also promote vascular repair, helping re-establish blood flow to ischemic or damaged brain regions. Some studies suggest that dairy-derived EVs upregulate VEGF and angiopoietin pathways, enhancing angiogenesis and capillary stability, a critical step in restoring nutrient and oxygen supply to healing neurons. Another intriguing role is their potential in modulating oxidative stress. The aftermath of brain injury involves a burst of reactive oxygen species, ROS, which damage mitochondria and impede recovery. Some of the peptides and miRNAs found in yogurt-derived EVs seem to increase the expression of endogenous antioxidant enzymes like SOD and catalase, while also supporting mitochondrial biogenesis. This could reduce secondary damage and improve energy availability for neural repair. Perhaps most fascinating is the potential to use yogurt EVs as natural nanocarriers for targeted drug delivery to the brain. Their intrinsic biocompatibility, ability to be orally administered, and resistance to enzymatic degradation in the GI tract make them ideal candidates for functionalized therapeutics. In theory, these vesicles could be engineered or loaded with neuroprotective compounds, such as peptides, anti-inflammatory agents, or even gene editing tools, and then directed across the BBB to sites of injury. In preclinical settings, dairy EVs have already been explored as carriers for curcumin and other neuroactive compounds, significantly enhancing their brain bioavailability. Of course, we're not yet at the stage of prescribing yogurt shakes for concussion recovery. Challenges remain, particularly in standardizing vesicle isolation, scaling production, and ensuring consistent therapeutic payloads. Human studies are still in their infancy, and the complexity of brain injuries varies dramatically between individuals. But what's striking is how this ancient food is revealing new capabilities, down to the nanoscale. The EVs in yogurt are not just leftovers from fermentation. They are biologically intelligent particles capable of crossing physiological boundaries, calming neuroinflammation, and possibly even helping the brain knit itself back together after trauma. If future research holds up, yogurt-derived EVs could become part of a new class of naturally sourced neurotherapeutics, affordable, biocompatible, and surprisingly effective. In the meantime, the next time you enjoy a cup of probiotic yogurt, consider this. Beneath its creamy surface lies a constellation of micro-messengers, and some of them may be quietly unlocking pathways to healing not just in the gut, but deep within the brain.